Hi, am I audible and is my screen visible? Is my screen visible to you? Um, okay, let me just once again. Okay, I will begin. If you have any problem in seeing the video or audio, let me know. Right. So thank you for joining the last session for this course. As I had mentioned previously, that this is the uh, the revision session for this course. So basically today what I'm trying to do is this is last session where we have so far we have discussed all the theoretical aspects that has been discovered so far in all the weeks. But today I will cover the questions or from different portion, basically the entire course. I didn't want it to be more of a theoretical one, or I didn't want it to be more of a where I'm describing the topics, because we have already done that. All my previous sessions are also based on these, where you can open those sessions and you can see those topics. So what I have done today is I have just given first few slides are just telling you has been the overall flow of the course, and after that I have questions from different assignments that you got in this semester or in this time of the course, and the solutions for them, right? And so the idea is that I'm giving you the answers. We have a lot of questions, and we will solve them as we go ahead. And to each question, I've given you the assignment number and the question number. And if you want to refer to the concepts which are associated with them, you will just have to refer to the week number. Let's say it's assignment 3. So in my session, which is problem solving session 3, that will have the relevant course materials for that, right? So I am not going to discuss extensively, but the agenda of today's session is just to give you a mind map for your exams or for the course in general, right? Okay, with that, let me begin. Yeah. So taking you back to the very first session that we had for this course, where we started by describing the macromolecules that are present in our body, right? So we have, or in a cell, so we have lipids, we have nucleic acids, we have carbohydrates and proteins, and we saw different types of each of these macromolecules. We saw how many different kind of lipids are there. We saw how many different kind of nucleic acids are there. We saw how many different kinds of carbohydrates. When I say different types, remember the classification of each of these macromolecules. In proteins, you have the way they are folded. So on the basis of their structure, also they are divided. So when you are looking into the basics of the macromolecules, be mindful of their name, their classification, and their like broad division, basically. So structural classification, numeral name, when you are giving the name. So when we discuss about carbohydrate, we extensively discuss something about RS naming system, DNL naming system for amino acids. So these are the particulars that you should be careful about, and these are not very difficult topics. They're easy. If you just see it once, it will be easier for you to understand. Just look into the how you are using the nomenclature for, right? So that was the very first few sessions that we did, where we looked into different kind of all these molecules, and then we dedicated some portion where we looked into each of these molecules, just the structure and the naming and how they go about it, right? Then we did extensively about enzymes, right? Remember, after all these macromolecules where we had looked into the classification, we went to enzyme, and the three maps, or the three yeah, the three figures that are on your slide right now, this one was basically to show you how enzymes are helping reaching or reducing the activation energy for the reactions. So we were looking into how reactions are taking place, and then we looked into how enzymes are taking place in or their role in this. Then what we did along with this was we looked into how this kinetics takes place. So we dedicated an extensive portion to enzyme kinetics. So when you are, if you are applying for exams, if you're giving exams, or in general, if you are revising the course, uh, when you come to enzyme kinetics, you be careful about this is the explanation for enzyme. Why do you have enzyme? Why do you need enzyme? And in enzyme kinetics, this is the most important curve. This is the Michaelis-Menten equation comes from this curve. So this is the same graph which is represented at straight line. And we know why that happens because there is Km. And we also know, so remember this map, this graph you should remember very, like properly. You should remember the slope is given by Km by Vmax. You should remember what is this point, what is this point, and all of these. Okay. So this equation is something that we discussed extensively. My suggestion would be, if you want to revise this topic, enzyme kinetics, try to solve more numericals. I have put 2-3 numericals if I'm not wrong. But you try more questions on Michaelis-Menten equation. That will give you a grasp of how do you play about with not just Vmax and not just Km. There is something called catalytic efficiency, so Kcat. Then you have total enzyme. How do you use total enzyme? There is derivation of this equation. So just look into those topics. They are, as you know, they are not difficult. We have sessions. We have several literature, several resources available to understand them. And my suggestion will be do more numericals on them, right? And in our problem solving sessions, we have done many questions which involve these equations, right? And after looking into kinetics, we did this 3 inhibition, right? So we did something called competitive, we did something called non-competitive, then we did something called uncompetitive, reversible inhibition, irreversible inhibition, and we had these graphs. This curve shows us that if you have competition between the enzyme and substrate and the inhibitor, then how does the kinetics pan out? So this is very important, and please be careful when you are looking into this, and this is important topic, so do revise this. What you need to know is, so this table basically summarizes what you need to know. You need to know the type of the inhibition, what is it acting on, and the effect on the three, or its effect on Vmax and Km, and how does that get overcome. Okay, how is it overcome? So you should remember this, that one is exclusively on the active side. So if it is an active side, it's basically the same side that is being competed by for the by substrate and the inhibitor. If it is not allostatic, it means enzyme can have substrate, but it can still bind with the inhibitor. And uncompetitive, it basically doesn't care about the enzyme's active site. It basically cares about the enzyme substrate complex. So with that, your graph changes, and with your graph, your equation also changes, and the way you're plotting the graph also changes, right? So this is easy to remember. When it is uncompetitive, it's parallel in line. But what you need to remember is how are the Km and the Vmax changing, right? I am not going to go into details because, of course, they are all there in the respective session and in your course. Okay. So once we did macromolecules, we did enzyme. In between, I haven't included in slides, but we also did pH. So we did a lot of equations and questions on pH calculation, molarity calculation. How are you calculating the pH? How are you calculating the H plus concentration? So you can look up to that also. Right. And then before we shifted our gears to catabolism and anabolism, we dedicated one entire problem solving session to bioenergetics, right? We looked at how delta G is changing and what is the equation for calculating the Gibbs free energy change, and what are the different parameters. We calculated the standard reduction potential. So I'm telling you all these terms which are things that we have discussed and which is pertinent to this course. So that is not part of today's, this is not in my slide, but I'm telling you before we jump. So why is it relevant? Because when you're jumping to these different pathways, you'll often encounter delta G. And with delta G, we know if the particular reaction is favorable or not favorable. If the lesser the negative the delta G, the more favorable or the more spontaneous the reaction is. So before you start all these breakdown and making of the molecules, just once look into bioenergetics and look how we calculate delta G and how do we there are 2-3 formulas for calculating delta G. So when are you using which ones? Be careful about that, right? Okay, and then we move to this particular topic, where the 4 macromolecules that we discussed, lipids, carbohydrate, proteins. So all these 4 here, lipids, carbohydrate, proteins, and nucleic acid. What we next did is, how is lipid broken down? How are lipids made? How is nucleic acid broken down? How are they made? How is carbohydrate is broken down, and how are they made, and how are similar to protein? So we looked into these 4 molecules, and we saw how are they built and how they are broken down. So all the catabolism and the anabolism steps, right? So we had glycolysis. The first thing that we discussed was glycolysis. We moved from glucose. The ultimate thing is to get energy. So we looked at from glycolysis, from glucose to pyruvate to what happens in several stages. And then we moved to citric acid cycle or the Krebs cycle, and we saw how does those changes take place. So this is in the flow. And after citric acid cycle, what you see is oxidative phosphorylation and electron transfer, basically ATP generation. So citric acid cycle, and then we had extensive discussion on how ATP synthesis takes place, right? This is by this collection here. And this, since we did oxidative phosphorylation, which is basically making you're basically in the process of citric acid cycle. So if you see the slide here, after the citric acid cycle, you have this water which is being formed, and you have ATP which is being synthesized after your glycolysis and citric acid cycle. But since this is oxidative phosphorylation, we did a similar concept after that, which is photophosphorylation, which is what happens in plants. In plants, they do photosynthesis, and we looked into how they do photosynthesis because that's the way they generate energy, and that's how we saw from photophosphorylation, how do you go to ATP? So first you do oxidative phosphorylation, then you do photophosphorylation. Okay. And then since we saw this, the next thing was we are breaking down. So we are all this while we are breaking down all the carbohydrates. The next step was how do carbohydrates are made. So we have seen how about carbon assimilation. And we also have seen about how glucose is made, how glucose is stored. So all this go hand in hand. So once we are done with carbohydrate, we move to amino acid, where we looked into amino acid catabolism in the form of urea cycle and the breakdown of amino acid. At the same time, we saw how amino acids are stored, how they are made, right? Then we move to lipids, and we saw how they are broken down and how they are made. So are either oxidation or how they are synthesized, right? So all these 4 macromolecules. So the basic theme of the course is those 4 macromolecules and everything related to them. Okay. And the last thing that we did yesterday was hormones, so which is also connected to lipids. So they are all interconnected topics, right? Okay. So that brings me to today's main part, which is discussing questions so that you get like grip of every topic. And the questions here are in no order. That's exactly how you will get it in any exams. So they're in no particular order. You have questions from all the assignments that we have received in this semester or this course this time. So all of them are dispersed, and we have a lot of questions, but we also have a lot of time to dedicate to them. So we will try to wrap up this question within 2 minutes so that we have ample time to cover all of these topics, right? Okay. Just a second. Yeah, let's quickly begin. Okay, so this is the first question, which is from carbohydrates. So some of the questions I haven't given explanation, but they are self-explanatory, but some of the questions do have the explanation of why the answers are there. So the whole intention of this session right now is this is a revision, and we want to solve as many as questions there are. And I chose the assignment questions so that they are something that you have just solved. So this should be very easy for you to go. If not, let's say you found out that your solutions were not correct, that's fine. We can just give it second attempt and see if it works out. Yeah, coming back to this. So the question is again, once we have discussed this question, I will tell you the patterns of the question that you are generally getting in this semester that you have seen. These were the topics that we'll be discussing the first time. First time we started these sessions, and your first week course syllabus covered carbohydrates, and we spoke about epimerization. We spoke about the types of saccharides that are there, types of carbohydrates that you have. uh, we discussed about keto sugar, we discussed about aldose sugar, then we also discussed about anomers, epimers, chirality. All of these are based on those part topics. So I'm waiting for the response, and then once we discuss the answer, then we can quickly jump on to the next one. So the answer says A, B, and D. Okay, we'll come to the answers. So for the first, let's just start discussing. So as I said, on the slide, you will find the assignment number and the question number that I'm referring to, right? So it's easier for you to track the the question. And if you want to have the relevant relevant topics in the problem solving session, you just need to follow the assignment number. Okay, right. Coming to the question, it says which of these are true for carbohydrates. So the first says D-glucose can be converted to D-galactose by epimerization, which is true. So they are epimers or C4 epimers. Remember, we discussed about one of the carbon being different. So that's epimers, and D-glucose and D-galactose are C4 epimers, right? So they can be converted to one another. Second one says maltose is an oligosaccharide formed by alpha 1,4 glycosidic linkages between two glucose molecules. That's true. So maltose is actually a disaccharide which is between two glucose molecules. Okay, two D-glucose molecules, and that's why what I meant was with this is, so D and L also you should take care of. So third one says glucose is a keto sugar with 6 carbon atoms, which was wrong. We had seen glucose was a 6 carbon atoms with which was an aldose sugar. It has a CHO group, right? And the carbonyl group, if you look into the glucose structure, is at C1, and the C1s then have CHO. If it had been in the C2 position, then you would have a double bonded O. So that would be a keto sugar, right? D is D-mannose and D-glucose are anomers. Again, D-mannose and D-glucose, if you remember, we had seen mannose and glucose are again epimers. They are C2 epimers. They are not anomers, right? So anomers are basically, if you remember, anomers are the ones that have, so if you look into their structure, they are structurally different at the chiral center. So if you see that in glucose and mannose, they are not different at that center. They are different at C2. Okay. And the last one says amylopectin is a polysaccharide of glucose with only alpha 1,4 linkage, which is wrong. It has, so the linear structure has alpha 1,4. They also have branch structure, which is alpha 1,6 linkage, right? Okay. Now, what I wanted to highlight is, I also mentioned this briefly yesterday. Most of the questions in this semester you would see are of this format: correct, incorrect. So these questions are very, in one way, they are very, very informative, because they in those 5 or 6 points, they basically cover a lot about the topic. So do attempt all these questions and try more playing with these questions. They will give, so for example, here only we have covered anomers, epimers, ketos, aldose. So these kind of questions are very helpful, right? Okay, quickly moving to the next question. Yeah, so this is about the remember I told about the lowest standard reduction potential. So this is one of those questions, and you are to select the one with the lowest standard reduction potential. So in these kind of question, my suggestion is these reactions that you see on the slides, they are very, very frequently we have encountered them, right? So why not just remember these at once. For example, NAD plus reduction is something that you have been encountering a lot. So remember these. Then you have cytochrome B. So we remember, right, reduction. So all these reduction that you see, they are we have ubiquinone, cytochrome C. You have seen in the electron transfer chain. So few of them you can just memorize because they have been quite frequent. Along with this, you have several other which are very frequent. So the frequent reactions that you see or the equation that you see in biochemistry, my suggestion is, if not the absolute value, at least remember the closest value to of these reduction potential. Okay, no issues. Because it's a revision session, we will just quickly look into the answer. So the answer, this one says the lowest standard reduction potential, and the answer is NADP reduction. And here I have given you the approximate. Again, I have told you, you don't have to remember the exact values, but you need to remember the, like the values which is close by, so approximate values. So remember these values, and as the question says the lowest standard reduction potential here is minus 0.32, which is of NAD plus reduction. So you can come back to this slide and look at the values. So the idea is that you remember these values, right? Okay, next one. Again, let's do it quickly. So these are basic questions about gel electrophoresis, and what you are to do is look into the correct answers. C, D, and E. Okay, so just a second. D and E, yes. We'll also look into by B and not C. Okay, so we are to look into the correct statements. So the first one says enzymes purified by SDS page retain their activity. This is an incorrect statement because if you remember, SDS page is a denaturing technique. So the SDS, which is sodium dodecyl sulfate, it basically is used to linearize, give a uniform negative charge on the proteins, right? So it unfolds the protein and give it an uniform negative charge, and then it is supposed to resolve in the gel, right? So if you are losing the native structure of the protein, then its activity is also lost. So the first statement is incorrect, that we had got right. Next one, it is suitable to determine the degree of purity of a protein in a mixture, right? So generally you will see that SDS page is used to assess the protein purity, which is based on the bands. So if you have only one single band, which is a very, very clear and sharp band, then you know that it's a very pure protein. If you have multiple bands, then you know that it is a mixture of proteins. And that's what you will generally see if people are resolving a protein. If they see multiple proteins, that means they have several different kind of proteins in that mixture. If it's just one clear band, that's one single protein. So that gives you an idea about the purity of your sample. Okay. Next one says in P page or in polyacrylamide gel electrophoresis with SDS, the protein migrate in proportion to their charge to mass ratio, right? But here, remember their charge to mass ratio will be equal, because they're all negatively charged, right? So in SDS page, they primarily migrate according to molecular weight, and that's what gives them the different bands, right? The charge is uniform because we have put a uniform negative charge using SDS. So not charge to mass, but in SDS page, it moves according to, or based on their size or the molecular weight, right? So that's why it's incorrect. Fourth one says SDS page can be used to separate the subunits of any multimeric protein. Yes, we just did that. And along with that, SDS, which disrupts non-covalent interaction and of course breaks it up into different units. So we can separate those units of multimeric protein. And the fifth one says that two protein with similar molecular mass can be separated based on isoelectric point. That's also correct, because that's the principle of isoelectric focusing. So if you have same mass, then you separate them based on their isoelectric charges, right? Or isoelectric points. So these are correct. So C is incorrect because it's based on the size, and B is correct because you do see multiple bands and you know about the purity of the sample, right? Okay, next one. This I remember we have discussed so much in the sessions. Basically, it talks about what is the molarity of the pure water at 25. This by now, you should remember this number directly. C, which is 55.5. Fantastic, because we have extensively discussed this, right? And let me just go over once again quickly so that people who are not able to solve can at least see the logic behind it, right? So the question says, what is the molarity of a pure water at 25 degrees centigrade, right? So molarity is, as we know, number of moles per solution of the solution per liter of the solution, moles of solute per liter of solution. So this is how we represent it, right? Now, when you say pure water, we know the density is 1 gram per ml or 1 kg per liter. So with that, we can calculate the mass of this water. So you have the density, you know the volume also, because you are given that it's a pure water. So you know it's a 1 kg or 1000 grams. So you have the mass. That means for molarity, you can find out the number of moles. So number of moles, as we know, is given by the mass of the solid, which is divided by their molar mass. So you have 1000 gram by 18 gram, 18 gram H2O, if you remember, 6 plus 16 plus 2. So 18, so 1000 by 18 is 55.56, and molarity is basically 55.56 mole by 1 liter, which is 55.5 molarity. We have done this and more similar questions. If you remember, we had this extensive numericals going on. So I'm glad that you remembered it. So good job there. Yes. Okay, next one. Now you have aldohaptose, and how many stereoisomers can you have from its linear form? Two to the power. Can you give me a digit for that? I mean, the number for that? What is the exact value? 32. You're doing great. Fantastic. Right? And the reasoning behind that is that if you look at the aldohaptose, so if you look into the structure of aldohaptose, this is how it looks: CH2OH, CHO, and these are the so 1, 2, 3, 4, 5, 5 carbon in between. Now, if you look into this carbon, this has double bond O and H, so this is not a chiral center. This has HOH, so again, this is not a chiral center. Let me remind you what is chiral center. It's basically a carbon with 4 different functional groups. Here you have 2H, here you are double bond with O. So you don't have 4 different groups. So these 2 are not chiral center. You have 5 remaining chiral center. If you look into this carbon, it has CHO, OH in one side, H in one side, and this entire remaining body in the other side. So it has 4 different groups on each side. So this is one chiral center. Similarly, 1, 2, 3, 4, 5. These 5 are chiral centers, and the chiral centers can be present in stereoisomers. So if there are 5 chiral centers, you have 2 to the power n chiral centers, which is basically stereoisomer. So 2 to the power 5 is 32 stereoisomer. So great job. Yeah, moving to the next one. Let's do this. Which basically says calculate the standard free energy change for a reaction at equilibrium where the concentration of the product is 4 times the reactant's, and the temperature is physiological temperature. So physiological temperature, if you remember, we just encountered these in the molarity of water question. So try to remember the different delta G formulas that we had encountered. How do we calculate for once in one of the just to give you a hint, in one of the formula we had only the reduction potential. In one of the formula, instead of with the reduction potential, you also have the relative concentration of reactants and products. So you have these 2 forms, and you remember we discussed different questions of when and how to use these. So that's basically the formula that you need to use in this question here. You are already told that you have a concentration of the product is 4 times that of the reactant. So using that particular information, you can use your this equation here. Okay, and also along with that, remember the questions will often give you in what you should report. Like for example, here it says your answer should be in kilojoule per mole, and if you remember, we had done in the problem solving sessions also, we converted the forms, and then we converted the what do you say, the units. But remember to come back to the unit that is asked in the question. Why I say so? Let's say we calculate something minus 300, minus 3 points, minus 300, minus 3000, but that's in, let's say, joule. So the question asks is kilojoule. So your answer should be 3 or 0.3. So be mindful of what you are answering, because the question tells you what kind of result you want. Yes, exactly. Right. So we've got an answer which is 3.57, which is what is given in your key, but you will get answer which is in the range of 3.45, like you have mentioned. So 3.45 is what the correct values are. So you will see the answer keep giving you minus 3.5, but your value should be near that, right? And we have used this formula, which is delta G is given by minus RT ln K. Ln K is basically your products by reactants. And we know the product is 4 times reactants here. So we have to simply do ln 4, which is 1.386. And then substitute this value into the formula. Delta G minus R. R we know is a gas constant. And that's why I told you that time also, just remember the value. It's 8.314. And T has been specified as physiological temperature, which is 25 degrees centigrade. And in Kelvin, it is 298 Kelvin, right? And then we substitute this with ln4 value, which is when you solve, you get in joule per mole. Why joule per mole? Because gas constant is in joule per mole. So do not get confused here. Your answer should be in joule per mole per Kelvin. But the question, if you look at the answer, which is the unit which is asked is kilojoule. So you basically convert this to kilojoule and report it, right? So yeah, that's about it. But yeah, we have done many more such questions in the problem solving session. So do just practice those questions once again, right? All right, coming to this. This is about the nitrogenase complex. Remember about the leg hemoglobin, and we discussed about how nitrogen fixation takes place and what are the bacteria that are taking part in this and why do you have this leg hemoglobin and all these. We discussed all of this, right? So this is for those questions. Okay. Just read the question once again. Leg hemoglobin, is it the only answer to this? Read it carefully. Can you have possibly other answers as well? Okay. That's it? Okay. Well, the answer is basically the entire set here. And this you would, in words, you would find this in the Leninger book, right? So if you look into the Leninger book, you will see that all of these have been, like touted to be a measure to overcome the toxicity of nitrogen complex in presence of oxygen, right? Remember, we had discussed how oxygen can be toxic for nitrogen complex, and that's why the plants have devised several ways to overcome the toxicity. And we that time we had discussed about leg hemoglobin, how A, B, C, D as well. So yeah, B, C definitely, but A also, and D, which we are also looking into D, right? And as I mentioned, the 4 of these sentences are as it is mentioned in the Leninger, which basically tells you all of these are a way that has been evolved to counter the oxygen toxicity. So the third one we all are very well aware of. The first one is basically in some of the bacteria, you will see that they have uncoupled the electron transfer and the ATP synthesis. So they don't do that simultaneously, right? So ATP transfer can take place and later it can do ATP synthesis, which is concomitant in the system that we have discussed. Next one is symbiotic relationship between leguminous plants and nitrogen fixing bacteria is basically the leg hemoglobin synthesis, right? The fourth one says availability of TCA cycle intermediate in the root nodules is basically more these nutrients are, or more these intermediates are available, more the activity of the complex is there. So these are also a way that you're concentrating the intermediates where the root nodules are there, so that you don't have these nitrogenous complex sitting. They are always active and they're using up, and they don't get to interact with the oxygen much more, right? Okay, the next one. So which of these pathways lead to compounds that can be used to synthesize glucose? So remember glucose pathway, what are you using for synthesizing? And remember, what are you doing here? What does these pathways lead to? B. Okay. E, D, and E. Okay. So let me tell you the answers. Right. So the answers are A, B, and D. Let me just quickly see what all did we get an answer as in A, B, D, and E. A, B, D, E. Okay, not maybe E, but yeah, I am seeing someone typing A, D, E, A. Okay, all right. No worries. Okay, so coming to it, the first one says breakdown of lactose by lactase. So if you have lactase breakdown, lactose breakdown, which is itself disaccharide composed of glucose and galactose. So if it breaks, it gives you glucose and galactose, and we know that galactose can be converted to glucose. So it is used to synthesize glucose. That's correct. Great job. Next one, catalysis of catabolism of tyrosine, leucine, and alanine, which is basically amino acid degradation. And we know that so alanine, for example, if you are so it can be converted to pyruvate, which can convert to glucose. We know the alanine pyruvate cycle very well. Tyrosine also is glucogenic, so it can and as well as ketogenic, so it can yield fumarate and acetoacetate that can also be used for glucose synthesis. Leucine also uh, can be used. So it can be used for glucose synthesis. That's why it's correct. Now, the third one, which I did view option. Okay, great. So you did not answer that, which was great. So ketone bodies, if you break it down, you do not get glucose because if you break down keto acids, they are derived from fatty acids, right? So if you break down ketone acids, you get acetyl coenzyme A, and acetyl coenzyme A cannot go back into glucose form. So if you remember glucose to pyruvate to acetyl coenzyme, so the reverse cannot take place, right? Now D, which is hydrolysis of TAG. If you hydrolyze TAG, you have glycerol, which can be converted to DHAP, and DHAP can be converted to glucose. We know that it's the reverse of glycolysis or the gluconeogenesis, right? So if you break down TAG, you get glycerol group, right? Now, however, when you break down cholesterol, which we did yesterday, if you break down cholesterol, they are forming bile acids and hormones. If you remember, they start from acid, but that doesn't mean that when you break down cholesterol, they will form uh, acetate. When you break down cholesterol, remember we did this also. They are used in the form of steroid hormones and bile acids. So they do not contribute to gluconeogenesis. So be mindful of this certain changes. Yes, it looks like that, okay, cholesterol is formed from acetate, so it will be reversing it. That doesn't happen actually, because when you break down cholesterol, you get bile acids and you get the hormones that you basically breakdown is basically you're using the cholesterols in different forms. So it doesn't mean that you will when you use it up, it becomes glucose or it becomes acetate for that matter, right? Okay, the next one. This is about coenzymes. So you have to look into this. So we have discussed about several coenzymes. So this question will give you a recap of what we discussed about coenzyme. You see, coenzyme A, we have discussed about biotin, we have discussed about NAD+, we know about riboflavin. So it just basically asks you what are false statements, remember, not true, false statements. Uh, Once again, these are all from your assignment questions. I'm pretty sure you would have already looked into these questions, but just so that it's a good revision. A, B, C, and someone said D. Okay, you have to give me the false questions, right? False is A, D, and E, right? So we'll see why. Now, why is the first statement wrong? Coenzyme A is associated with the transfer of alkyl groups. Um, if you remember fatty acyl coenzyme A, that should tell you that what it is involved in. It is involved in the transfer of acyl group and not alkyl group. So A is false. So we did get A as an answer. Yes. Okay. Next one is biotin is involved in the covalent transfer of CO2 between two molecules. Yes, that we know. And we did, where did we see this? We saw in the acetyl coenzyme A carboxylase, if you remember. In that pathway, we had seen biotin as a CO2 carrier because that's how carboxylase, you have the carbon being added, right? Next one is NAD plus K plays a key role in ETC. We have seen this, that they are part of the electron transport chain, right? Uh, Next is coenzyme A plays a major role in glycolytic pathway. If you remember, you do not see coenzyme A in any directly in any glycolysis step. Where you see it actually in the glycolysis is after the step, which is right after the glycolysis step, which is basically pyruvate getting converted to acetyl coenzyme A. That's where you see coenzyme A, if you remember. You have the acetyl pyruvate, which is getting converted to acetyl coenzyme A. So there you have your coenzyme A playing a greater role, right? And the last one is riboflavin is involved in amino group transfer. Uh, Basically, it is not correct because riboflavin is giving rise to FND and FMN, which are involved in oxidation and reduction. If you remember, we did this in the ETC where we saw into these electron carrier where we saw FAD and FMN. So you have riboflavin giving rise to the FAD and FMN. For the amino group transfer, we had PLP, if you remember, pyridoxal phosphate. So these are names that you should remember: TPP, biotin, riboflavin, coenzyme A, NAD plus, FAD plus, NADPH. So all these you should remember where are they used. So be careful when you are revising which comes where. Okay. All right. Quickly. Next one. Okay. Very interesting question. We have already seen this once. So in the overall reaction of beta oxidation cycle, which is in your fatty acid that this was discussed in fatty acids. So there in beta oxidation cycles, then those compounds are taken further for TC and ETC. So if you take the overall reaction, this is how it looks. Palmitoyl coenzyme A converts to coenzyme A, gives you ATP, CO2, and H2O. You are to give me the stoichiometry of ABC as sum of these three. So take your time, think what is the correct value, and then just give the answer to this. So when we were discussing our oxidation and we were fatty acid oxidation and we looked into palmitoyl coenzyme A, this is exactly the last equation that I had shown in the like in the slide. So this is exactly from the book or from your notes. Okay. So I got one response. Okay. I will wait for the other. Okay, very nearby answer. The answer is 147. And this is what we discussed, if you remember. We had palmitoyl coenzyme A. The result from your beta oxidation is palmitoyl coenzyme A using 7 coenzyme A, oxygen, and ADP and PI to give you 8 acetyl coenzyme A, 28 ATP, and 7 H2O. Right? Now, when you take this acetyl coenzyme A, further, for your TCA and ETC, you get 8 coenzyme A, 80 ATP, and 16 CO2 and 16 H2O. If you look into the stoichiometry and think about all these different, this is what I have been telling. I think I repeatedly told in the first few sessions. Be very careful of how many ATPs are being synthesized and how many CO2s and how many. So you will get stoichiometric question. That is one of the very important aspect about such pathways, right? So your acetyl coenzyme A, this has 8 acetyl coenzyme A, gives you this. So if you add them up together, you basically have 108 ATP, this 80 and this 28. This 80 comes from this acetyl coenzyme A. So and 108 ATP, 16 CO2, which comes from the acetyl coenzyme A getting reduced in the GCA cycle. And then you have 16 H2O here and 7 H2O from the first, from the beta oxygen step. So overall, you have A is 108, B is 16, and C is 23. A plus B plus C is 147. So you can check where you lost out the 2 ATPs that you, the 2 difference that you have for respect to 145. The values are here on the slide. And we have discussed this when we were doing the problem solving session, right? Okay. Let's move to the next question because we have a lot of other questions as well. And this is one of the questions that we, I think, spent at least 1-2 sessions clearly on this. So you have pH of the solution, which is 3, and you have to tell me the H plus concentration, which we know is given by 10 to the power minus n. So just let me know what is the n. By now, this question should be like very easy for you. They are very easy questions. Just read the question once again, maybe. Just read the question. What exactly are they asking? H plus or base? Please don't miss out on the keywords. You are getting there, but just read it. It says conjugate base or conjugate acid. Okay, let me give you the answer. This is also something that we had discussed in the class. N is 11, okay? And this is what we are doing here. The question says if pH of an HCl solution is 3, which is correct, you guys are correct. The H plus concentration in its conjugate base is 10 to the power main. So you're basically we're trying to look into the conjugate base here, right? Because we already know the pH. So pH is given to us. We know the pH, we know the H plus concentration. We are trying to find the conjugate base here. So your Kw, if you remember Kw here, we had discussed Kw is basically the ion product, which is 1 into 10 to the power minus 14, right? And Kw or ion product is basically your H3O plus and OH minus. Now from there, H3O plus, we can calculate from the pH itself. We know what is H3O plus, right? We need to calculate the OH minus. So OH minus is basically just the reverse of it. Kw by your H3O plus. This is exactly what you people just did. pH 10 to the power minus pH gives you the concentration of hydronium ions. So that is what you did. 10 to the power minus 3. But what you are asking is 10 to the power minus 8 of its conjugate base. So OH minus is equal to 10 to the power minus 11. And one more thing, while you are answering both, so I saw you wrote N is equal to minus 3. We're asking n. So you do not write minus because minus n is what you are answering. Like what I meant is the answer should be absolute terms here. So let's say minus, and then you write minus of minus 3. So it becomes plus 3. So it's 10 to the power plus 3. That's not what it is, right? Minus n. So minus sign you can avoid. We are asking you just about the n. So that's what. Be very careful about what the question is asking you. If it's just asking n, just report the n values. Let's say you had minus 3 and minus 4 as the answers, but your answer should have been 3 and 4, not minus 3. Because I'm asking you n. So the formula says 10 to the power minus n. Now you are giving me n as minus 3. So in the formula, it will become 10 to the power minus of minus 3, which is plus 3. So 10 to the power 3. That becomes something else completely, right? So be very careful of what the question is asking. Okay, that's my only advice to you here. And the question we have already discussed in this class also. So you can go back to that problem solving session and look into this, right? Okay, next one. So we have squalene synthesis, and this was in your assignment, of course, the last assignment. And it says squalene synthesis from acetyl coenzyme A utilizes which of these compounds? Or what are the steps there? So I told you last class also, I mean last to last class, that we have to look into squalene synthesis also. So do read up about isoprene, squalene, all these synthesis. Okay, I've got one response. Wait for 10 seconds so the other person also can respond. Okay, let me give the answer. We have to move to the next questions as well. Right, so the answers, correct ones are actually A, B, C, right? If you remember the squalene synthesis pathway, we were starting with mevalonate, remember? So you had mevalonate, which was formed from 3-acetyl coenzyme A, and then from mevalonate converted to IPP, and then the IPP, 6 of those IPP form the squalene, right? And then you had farnesyl pyrophosphate. Remember all these things that we had discussed. I'm not going into the details of it. ABC, yeah, exactly. That's the answer, correct. So if you remember this steps, then the correct ones are, as I just discussed, 6 IPP molecules, 4 NADPH, because you have 2 NADPH during mevalonate synthesis and 2 NADPH, which is during farnesyl to squalene synthesis. That's there. Third one is 9 phosphoryl transfer reaction. So you will see that you have, when you are generating one IPP, if you remember there, you have 3 ATPs. And overall, we are using, like 6 IPP is being generated. So you have 18 phosphoryl transfers, right, overall. But when you have IPP, it's basically getting isomerized. So 3 IPPs and 3 DMAPs are used. So there you have 9 ATPs, which you will be using now, right? So the correct answer is ABC. And as I said, if 4 NDPH is correct, then 13 is incorrect. And you don't have 18 ATP because not 18, but you have 9 ATPs. If you look into these steps, so whenever I told you have been repeating, look into the ATPs of each of these steps, right? Okay, next. This one is very, very simple. So this is like the basic question that is being asked. So this should be easy for you to do. So the question is basically I'm sorry, I don't know why both of them show the same question number. I will change it later. So in your left portion, you have to match the structure and the name. And on your right, you have to tell me which glycosidic linkages are used in glycogen. A, B, C, D. What is ABCD? For which one are you talking about? Oh, wait, I will just give you the correct question numbers here. So this one is 2 and this one is 3. So you can answer accordingly. For question number 2, what is the correct answer? And for question number 3, which one is the correct answer? Yeah. Yeah. Right. Now you can answer it clearly so that you also don't get confused. AD14. AD14. Okay. I understood what you say. 3 is D. Okay. That's it. Let's move quickly. So 2 is D and E. Did we get that? Okay, let me tell you. And 3 is D and B also, right? You also have 1, 6. So look into the structure of glycogen. You will see this one. And here you have B as 3. So B is for TAG, triacylglycerol. And C, sorry, let me do it first like this. B is 3. B is 3. C is 1. See, if this structure is for deoxyribose. Okay, because your A is for ribose. So this is ribose and this is deoxyribose. Why? Because you don't have oxygen in the second position. That's the idea here. Deoxyribose has no oxygen. So deoxyribose, right? This is ribose. If you remove OH from here, it becomes deoxyribose. And then you have phosphate here, it becomes phospholipid. If you don't have this, it's just triacylglycerol, right? And this one has alpha 1,4 and alpha 1,6 if you look into the structure. So be mindful of that. Okay. Another quick one question. I told you about enzyme also will do. So one question on enzyme. So just to give you what do you say? Revision of Michaelis-Menten equation. The question is, for a given enzyme catalyzed reaction, the Michaelis constant, try to remember where we use it, is 0.6 millimole, and substrate concentration is 1 millimole. So what is the fractional saturation of the enzyme under these conditions? Try to solve it. If you can't, it's okay. It's anywhere revision class. So I will give you the formula that you have to use here. Okay. Because we are short on time, I will maybe wait for 10 more seconds and then we go ahead with the answer, because I do have a lot of questions and I want at least you to see the questions. These are from your assignment, yes, but just to see the questions. Just in case we are not able to complete it in the time that is the 2 hours that we have, what I will do is you can leave after 8 if you have some work to do. If we don't complete, I will show the questions and you can come back for the recording and see the questions there. Okay. Yes. Yes. Right. I've got the answer and which are both correct. Yeah. So the answer is 0.625 or 62.5, like you mentioned, both of them are correct. Because I haven't specified here in what terms I want. So you can answer in any way that you want. And since you have got the correct answer, I'm guessing you did exactly what is given on the slide. So just for the people who are there, if they join later. So V is equal to Vmax by S, Vmax into S by Km plus S is what we know as the Michaelis-Menten equation. And at the same time, we also know V naught is equal to K cat into ES and V max is equal to K cat into ET, all the enzymes being used. So what have we said? That we have Michaelis constant as 0.6 and we know the substrate concentration is 1 millimolar. And what I need to know is what is the fractional saturation, how much of the enzyme has been used. So what I'm supposed to do is what is the enzyme substrate complex as compared to the ET, right? So basically I'm supposed to find out what is the V naught as compared to the Vmax that can be possibly reached. So when I substitute these values here, I get S by Km plus S. If you resubstitute the equation for these values, you will get, and everything gets cancelled out. You have only S, Km by Km plus S, and these values we are given already 1 by 1.1, 1.6, which turns out to be 0.625 and 62.5. And like I mentioned, because no specific unit is given, you can write both the answers. Fraction is in both senses, right? Great job. Okay, quickly. You will have these match the following questions often, which is a good revision for us to know the names and where they are found and what type of the lipids you can find. So there are matches already made. You need to point out to the correct options here. So basically you have type of lipid, storage lipid, membrane lipids, or them having either phosphatidic acid as their functional group or sulfate group. Second one is what is the type of, so these ones on this side says the functions and the structure. This second column tells you about the type or name that is given to them. So glycolipid, phospholipids, TAGs or archibacterial lipids. And on the extreme right, which is in 1, 2, 3, 4, you have the specific names for these type of substitution. So let's say glycolipids, you have this group, what name does that particular lipid get? This is what the entire form is, and you need to look into the combinations that are given to you and give me the correct ones. B382, B382. Okay, and one is saying D. Well, A, D, and E are the correct answers, right? So 2A, which is basically 2 is membrane lipid, 2 glycolipid, which is also sphingolipid. If you remember, they are incorporated into those plasma membrane. That's why this option is correct. 2A2, not 3A2, not sulfate group. If you look into the membrane lipids, they are glycolipids and they are sphingolipids, right? 1A1 is basically this, which is not correct, right? 2D4, membrane lipids, TAG, yes, again. Sorry, membrane lipids, archibacterial lipids, which are the diphthenyl diether. I remember doing this question in the session, problem solving session also. So maybe this question was there in the problem solving session as well. And 4B1, which is phosphatidic acid, 2 phospholipids because phosphate groups, and which are in the glycerol phosphates. So a good reminder for you or a good revision for you, but also go back to your notes and do check what are these, each of these structures look like, right? Quickly. Very, very simple question. We have also done this in the sessions, problem solving sessions. So the question is, first question on the slide is, what is the concentration of H plus in a solution, which is 0.1 molar NaOH? And the second one says, what is the concentration of OH minus in a solution, which has an H plus concentration of 1.3 into 10 to the power minus 4? So in both of them, in one, you have the concentration of the base. So you have to tell me the concentration of H plus. And in the other, you have H plus concentration. You have to give me the concentration of the OH minus. Same formula, both the side. You just need to calculate and tell me the value. Okay. Um, Okay. All right. Okay. 20 seconds, 10, 20 seconds more, and we can look into the answer. Yeah, sorry. 20 seconds and for the second answer, and we can look into the answer for these questions. Since we have to move to the next question, otherwise I would have waited for the answers. 10 more seconds. I am getting the answer, so let me wait for it, and then I will give the answers. So it's the same formula that has been used in both these distinct questions. And if you look into the formula, no, this just okay. These are the answers. This formula that you see here, which is the iron product formula, please just remember that, this is very important formula and you need to remember this, the value of Kw and the equation. If you do remember this, all these type of questions become very easy, right? I told you the same question, the same equation, Kw iron product being H plus concentration into OH minus concentration. And the value of Kw always stays constant. We know that. That's 1 into 10 to the power minus 14. That's why, if you remember, when we tried to find at what concentration, at what pH both of these are similar, you will find pH is 7, right? We found out that also. How did we came to that number? So be mindful, and once you have this formula, you just need to substitute the values and you will get the answers. One is 10 to the power minus 13, and one is 7 into 7, 10 to the power minus 11. So my my cue to you is, remember the over H plus and OH together will be 10 to the power minus 14. So if it is 0.1, it's basically 10 to the power minus 1. So to make 20 10 to the power minus 14, it will be 10 to the power minus 13. Just remember it like this. Similarly here, 10 to the power minus 4. So the OH has to be 10 to the power 10. But when you look into the division here, 1 by 1.3, when you adjust the decimals, it will come 10 to the power minus 11. So what I meant is, when you are looking into the question, it will become apparent to you what should be the answer, right? All right. Very, very interesting question, and we have already done the exercise on this. So just a reminder that these kind of questions also will come. Okay. Since we have a lot of questions after this also to discuss, so I will just quickly go to the answers. It's basically, if you remember the order. So remember this order of order of priority while you are naming using RS system. So OCH2, OH, NH2, COH, CHO, CH2OH, CH3, and H. And when you look into this order, these are the correct options. So remember this order, okay? That's very important. That this is based on RS naming system. So you remember we had this, where do we start and how do we go? This is that order. So if you look into RS naming system, this is the order that you need to remember. Okay. All right. Next one. Yeah, let's do this. This is about enzyme. Again, I remember I said you that these kind of questions will become very apparent and are good questions for you to get grasp of the topics that are subparts of the or the overall thing theme that you are talking about. So this one here is about enzymes. So we need to remember what are these what are the features of enzymes. This one is easy, right? You are just supposed to tell me which of these are true for enzymes. Okay. So for the remaining questions, I will wait for 1 hour, 1 minute for each of these questions and then give you the answers. So just so that we can move on with the questions that are there, right? You can try to solve in 1 minute. That also might help you in increasing your speed. But in case you don't get it, that's all right. What I am trying here is to discuss all the possible questions. And when you are looking at the recording, you can stop and again attempt it again, right? So CDE, which is exactly what is correct. So the first one says that each enzyme has a three-part number, which is incorrect. If you remember the naming of the or classification of the enzyme, they have four parts, something like EC 2.7, 0.1, something like this. So you will have four parts to it, right? The second one says enzymes can increase the rate of reaction by varying the equilibrium constant. This is a very, very basic of enzyme kinetics. They don't affect the equilibrium constant. They lower the activation energy. That's what they do, right? And the remaining three statements are correct. So cofactor generally are we refer to the inorganic moiety required for the enzyme activity. That's the correct statement. So we say the Mg2 plus or the Fe2 plus, these are the cofactors, right? So they are the inorganic moiety. And not every enzyme you see requires a cofactor or a coenzyme. Coenzyme are basically the uh, organic moiety. So not all the enzymes are required in both of these together at the same time. And last one says the apoenzyme is it's only composed of the polypeptide chain, which is also correct, which is basically just the protein part of the enzyme, right? And when you have the cofactor and coenzyme, it's the holoenzyme, right? So this is the basic nomenclature that you should remember. Again, we have matching pairs. So you have to tell me the correct pairs, and 1 minute again we have for each of these. Okay, 10 more seconds. Okay, so the answer to this question is B and C being correct. So the B one says carbon monoxide is uh, basically we are looking at the inhibitors, or the substances that inhibit that step to occur, right? So carbon monoxide stops the electron transfer to oxygen, right? B, which is antimycin, it is for inhibiting the electron transfer for complex 3 when you look into the ETC. And C is for 4, which is rotenone, which inhibits the FES centers transfer from the FES center to ubiquinone. And at the same time, the C, which is antimycin, it is also therefore inhibiting the electron transfer to oxygen. C, which is rotenone, is already 15-4, and D, which is cyanide, which is involved in the inhibition of electron transfer in the like overall electron transfer is again stopped, right? So you will see these two are correct options, and then respectively the others are incorrect. Yes, you can type your question, because most of the time I am on the other screen where I am trying to look into the question. So I will keep coming back to your chat box, but you can type your question. I can read and then, like solve, or you can unmute yourself and ask if you have any doubt. Okay. Uh, you can type or you can unmute anytime you want. I will go to the next question, and if you have any doubt, you can let me know. Right? Yeah, we'll do this in the meanwhile. And yes, like I said, I will just wait for 1 minute so that we are able to cover most of the questions that are there. Okay, I'm so sorry, but I have to go to the questions, and 1 minute is over. So let me give you the answers. And what you could do in the meanwhile is you can check if you were thinking in the correct directions, right? So the answer is 6.9. Let me read out the question first. The question says that there is a basic amino acid with an amino group in its R chain, right? So there is an amino group in the R chain, and the pKa for its COOH group is 1.9, and for its NH3 is 8.7, and for its R is 5.1. And I am to calculate the pi for this amino acid, which comes as 6.9. So in a basic amino acid, we have discussed this. The Zwitter ion, it loses the proton from the side chains as pH increases. So for me, the two pKa's which are important are the ones of the R chain and of the NH3 plus, right? And so when I'm calculating the pi, I will take the average of these two values, right? So when we discussed about the how do we calculate PIs, we had seen when to consider which pH for or pKa for considering the overall pi of the amino acid. So here, since it's a basic amino acid, we lose the proton from the side chain when your pH increases. So we take the higher one for 8.7 and we take the pKa of the R chain here, right? Not the COH terminal. Okay, right. Next one. This is also an easy question. So it is basically saying in the mitochondria, the first amino group that enters the urea cycle is derived from the reaction of which enzyme. So which enzyme is part of that reaction, the first reaction? No, it's okay. You can revise the urea cycle and you will remember we had discussed about glutamate dehydrogenase and then glutaminase. So these are the this is the enzyme that is important for the first step. Just a revision step. It's okay if you don't remember right now. Okay, let's come to this. So among the options given below, which one will have the highest free energy change? So the key here is standard free energy change. So you need to remember the standard conditions and then calculate the values for delta G and try to remember where we are using these values. It's very simple. K we know already. We know we have just solved an equation like this. So we are just trying to understand the different values there. We have already used the equation once in this session. B. Yes, exactly. And these are the values for each of them. So if you look into K equilibrium as 11 at standard condition, which is the standard condition, pH being 7, all the substrate and product concentration being 1 molar and 25 degrees centigrade. So if you replace it by K equilibrium as 11, this is the delta G. Then if you replace your concentration of product and reactants at 5/2, this is what the energy change is. If it is at physiological temperature and not at standard conditions, so what is the value? And respectively, you change the other. This is the change in the value. And the question is, which one will have the highest standard free energy change? So we know minus 2.2 is the highest. Right? Great job. Next one. Now, which one of these statements, which is related, this is related to photosynthesis, is correct? These statements are actually quite easy. So you just need to find out the correct ones. The incorrect ones are in fact very prominent. So it becomes very easy. B and A, and E as well. Okay. So I believe you would have come to that. But yes, like you have already mentioned, the first one is correct. Exciton transfer is a form of energy transfer in photosynthesis. We remember, right? We saw how there was transfer of excitation energy from one pigment to molecule to another pigment molecule in the light harvesting complex. So we saw that scheme and how they're transferred, right? Then second is chlorophyll. In chlorophyll, you have Mg2 plus, which is coordinated to nitrogen. That is also correct. C says that chlorophyll A and B differ only in their phytol side chain. So if you look into the structure, they are basically they have the same phytol tail, but they differ in their porphyrin ring position. Okay. So what do they have in their porphyrin ring? What is present in the porphyrin ring? One has a methyl group and one has an formyl group, right? D says chlorophyll D is not present in green plants, which is absolutely not correct. We know chlorophyll B is present. Okay. And the fifth one is correct, which is saying that biphycobilins are the primary LHCs or light harvesting structures in cyanobacteria. That is correct. Next one, which is again very simple. You need to tell me which one is correct and which one, sorry, what is the value of the two pH changes that you see? The question is, if you have pH 5, then what is the H plus concentration? And if you have pH 7, then what is the H plus concentration? And how much times do you have the other one higher than the next one? 100. Fantastic. That's great. This is exactly what is the case here. So you're trying to calculate 10 to the power 5 minus 10 to the power 7, which is 10 to the power minus 2, which is 0.01, which is 100 times higher in pH 5. Very, very well done. Okay, next one. Set of questions which again refer to statements. This is from bioenergetics part. So this, as I mentioned, these are from bioenergetic section, which we have discussed in one of the problem solving sessions. And this you have to tell me the correct statements. You're telling me the correct or you're telling me the incorrect? Remember which answers are you giving to me? Okay, so A and C, well, those are the only two which are incorrect. Okay, the others are correct. It's okay, it's not a problem. We'll see why. The first one says due to their large negative delta G, hydrolysis of molecules like ATP and PEP does not require a catalyst. Remember, we discussed this that they are thermodynamically favorable, but that does not mean that it will take place spontaneously. It is kinetically slow, and that's why you need enzymes, right? So you need a catalyst that brings down that activation energy. Okay. Second one is correct. The product of hydrolysis of PEP is more stable than the reactants. So when you hydrolyze PEP, you get phosphoenol pyruvate, you get pyruvate and inorganic phosphate, which are more stable due to resonance and tautomerization. So these products are more stable than PEP. Third one, which says resonance stabilization of the reactant promotes forward reaction. Uh, so uh, not the reactants, but resonance stabilization of the product. Because when the product is stabilized, it is when the reaction will take place, right? So resonance stabilization of the product, right? Fourth one says a high negative standard free energy change is a result of electrostatic repulsion in reactant. That is exactly what the underlying principle is. And fifth one, ionization of the product of ATP hydrolysis contributes to the stabilization. Again, the same thing that we have been discussing so far. Okay. So did you mention the correct or did you mention the incorrect? But that's okay. If you mention, if you were trying to answer the incorrect, you were correct. Otherwise, it's okay. It was not. It was just be mindful of what you are looking into. Okay, very quickly this one. This is again. Remember uh, the Michaelis-Menten equation, and from there you remember the Lineweaver-Burke equation plot, and then remember what are these points referring to. If you have that with you, you know what to do. A good question. In fact, a very interesting question. A. Okay. Anything else? E. Okay. Well, D is the correct answer, not E, and we'll see why. Like I said, look into this and look into this. So wherever the intercept is there, that is the intercept on the y-axis is 1 by Vmax. Intercept on the x-axis is minus 1 by Km. So this you should remember. Okay, and it's easy, right? This intercept is 0.1, this is 0.5. And for ET, remember the K cat is equal to Vmax by ET, right? So when you know that Vmax is either for 2 or 4, and we are seeing here that 1 by Vmax is 0.5. So when you inverse it, it becomes 2. So that is correct. Next, you are told that you have your ET as this and turnover rate, which is basically K cat. So you need to find out the K cat and uh, given your ET is there. So you need to just replace Vmax by VT, and we have already established Vmax is 2, and 2 by 2 is 1, and 2 by 2 is not 4. So this is correct answer. This is minus 1 by Km is minus 0.1. So Km is the inverse of it, right? Interesting question and a good revision. Okay, we'll do this quickly. This is very easy actually. Quickly, we'll do this question so that we are able to cover the other questions as well. What is the oxygen state of carbon in acetone? So remember the acetone structure and give me the oxidation state. This is something that we discussed in the very first few sessions. Right. Plus 2, because this is how your carbon is, or the structure is, CH3COCH3. And as we know, each of the CC bond, you have equal sharing. So there is no gaining of electron, or there is no pull of electron. But in the CO bond, because oxygen is more electronegative, carbon will lose the electrons. So that will give rise to +2 as its oxygen state. Right? Great. Let's do these questions, which is again on bioenergetics. Bioenergetics was something that we had discussed a lot of times. So again, the more you solve the question, the more you get well versed with the topics. So as you see here, when I say bioenergetics, you're basically discussing about the delta G of each of these reactions, and you see the reactions are from the citric acid cycle. So it also gives you a revision of the citric acid cycle. We still have some 10 questions to go. But yes, if you after 8 o'clock, if you need to leave, you can leave. But if you stay back, then it's okay. We can just discuss. Otherwise, it's also fine if you want to leave. Okay. 20 seconds, and then I can discuss these answers. So as you saw, most of these questions are from assignment, from the assignment that you received this time. So these are the answers for those questions. And some of the questions were numericals, which we had already discussed in the respective sessions. So you would also find more of these questions in those sessions. Okay. All right. Because we have to move on, I'll give the answers for this question. But what you can do is when you are doing on your own, you can come back and pause this question, give your answers, and then look for the answers again. Okay. And meanwhile, you can keep commenting on the chat box, whatever you feel is correct. Right. So incorrect statements are C and D. And the first one says delta G for the conversion of succinate to fumarate is 0 is correct. Second one is the electron transfer from succinate to fumarate is in the form of hydrogen. That's also correct, because in this reaction where you have succinate to fumarate, you have 2 hydrogen getting removed. So it is correct. Third, which says succinate dehydrogenase is present in the periplasm of prokaryote. That is incorrect, because this is present in the inner membrane of cytoplasm. Okay, not on the periplasm, not in the intermembrane space. And the last one says fumarate does distinguish between L-malate and D-malate in the reverse reaction to synthesize fumarate. That is incorrect, because they are very stereospecific. They need L-malate to convert synthesized fumarate, right? So they are these kind of B is correct. B is correct. Yeah, B is correct. Exactly, that's what. So again, before leaving, again, I will want you to give you one small hint, advice. When you are answering, cross-check your question once again. What does your question actually demands? Okay, right. Okay, this one quickly. Can you discuss the last question of 12th assignment? Okay. So I'll keep showing questions. In the meanwhile, I'll just check those questions in the assignment. Yeah, just a second. in the meanwhile, you can look into these questions. Just let me find out that question once from that. Yeah, I have found out this question. While I go to that question, I will quickly leave you with this one here. The correct answer here is C. So which basically tells the first statement is true, actually, because the first three steps of beta oxidation are basically like the conversion of succinate to oxaloacetate in terms of, you know, uh, how do I say that they have the dehydrogenation, hydration, and dehydrogenation, which is similar to what happens in TCA cycle. So that's correct. Rest everything is false because the second statement, which is conversion of hydroxyacyl to ketoacyl, is basically using NAD plus and not FAD. Third one says it is acting on cis enol or it produces cis enol coenzyme, and we know it is not the case, it is trans enol. And the fourth one says the second step consists of dehydration, which is incorrect. We know it is hydration, right? So that's why it is C, which is the correct answer. Okay, let's look into this one. So you keep answering these. I will just try to incorporate that question here. Okay. Okay. Just a second. So for some reason, I'm not able to, yeah, B, C, D, right? Well, first, let me just show you this question first. Sorry. Give me a second. I don't know why it got stuck here. Yeah, BCD is the correct statement. I will tell you about this question and then I will come to the question that you asked. Okay. Right. So in this, it says that all allostatic modulators are competitive inhibitors. Incorrect, right? We know that they don't bind to the active site. That's why they're allostatic, right? The second one says allostatic modulators are uncompetitive inhibitors, mediate conformational change in the enzyme. That's correct. Allostatic enzymes have specific pre-existing binding sites. That's also correct because they're not binding for the active site. Allostatic enzymes do not follow Michaelis-Menten's equation. Yes, because that's why they are changing the uh, kinetics in a sigmoidal manner. There is a saturation to it. And allostatic enzymes have hyperbolic? No. So like I said, it has sigmoidal kinetics, not hyperbolic, right? Which is the characteristic of Michaelis-Menten kinetics. Okay, before I jump to the question, were you asking about the tropic hormones question? Like basically you have a control and a test group of tropic hormones. Is that the question you were referring to? For some reason, I'm not able to share my screen in the other this thing. I don't know why. But okay, let me tell you the question, right? So let me read out the question to you once again. So I can see on my screen. You can open it in wherever you have the questions available to you, or just a second maybe. I will try to share that screen. Just a second, okay? Is this the question? The question is visible to you, right? Sorry. The question is visible to you, right? Huh. So the question says, following are the hormone levels in mice exposed to control and different compound blocking different endocrine pathway components. Now, based on the graph, identify the component that only affects the synthesis of tropic hormones. So what your idea should be here is, you need to know what is tropic hormone. Remember what is tropic hormone from our yesterday's discussion. Tropic hormones are basically those hormones that act on other glands. They are not the ones secreting the hormones, the final hormones. They are the hormones that release, they make sure that they and respective gland release those hormones, secrete those hormones. So where do these hormones come from? They come from the anterior pituitary, right? So what is the question saying? The question is saying that these are different compounds that are blocking endocrine pathways. Now, based on the graph, which is from A to C, so A, B, C, identify the compound that only affects B, A, C, whichever, only affects the synthesis of tropic hormones. So on your axis, you can see you have LH, luteinizing hormone, prolactin, aldosterone, oxytocin, insulin, testosterone, and HCTS. Now, what you need to do is, how you should solve it is, you should look into the tropic hormones, the hormones that are, the hormones that are tropic hormones. So out of these given hormones, which are tropic hormones, and then you should compare the levels of those tropic hormones. Let's say, for example, let's say the blue one. You have oxytocin level, which is almost similar in control and test group, right? So what you should see is which one is getting reduced. So it affects the synthesis. So your tropic hormone level should be reduced in test. Whichever tropic hormones are less in respective compound is your correct answer. Did you get the flow? So I cannot give you the correct answer because this is not released yet, but I can walk you through it, right? Just a second. So you have luteinizing hormone. Its value you should see, it's actually increased. It's actually here, the first one here, and so it is decreasing. This one is not decreasing. This one is decreasing to a greater extent, right? Now, similarly, you should compare the orange bar for the first and the second and the third. And first identify the tropic hormones and then compare them across the compounds. That is what you're supposed to do. Okay. All right. Wait a second. I will just quickly come back to the questions. Yes. Right. So I do realize that the time is over. So you can leave, like I have told you already, because the time is up. But if you want, if you can spare some moment, I would like to discuss, quickly go over the questions. You know, so that you know the questions. Maybe I can skip showing the answers. Quickly, I will show the answers, but I wanted to know the type of question. Give me another 5 minutes and I should be able to complete it. Okay. So what you can do is, yeah. In case you want to leave, that's fine. I would just like to wish, I mean, if you want to leave, that's okay. I will then discuss in the full form manner that I have to. Otherwise, I can quickly show you the questions. I'm just waiting for your response so that I know that if I have to start or not. In the meanwhile, I can just leave you with the next question. Okay, sure. So just give me a few more minutes. I think we should be able to complete. It's not that difficult. So the next kind of question is that you have 12 molecules of carbon dioxide incorporated into the Calvin cycle. So how many of these molecules will enter regeneration step? Right. So what you need to remember is the carbon assimilation Calvin cycle graph, and try to remember how many. So per carbon dioxide, how many of the molecules actually go into regeneration? Okay, so just let me give you a quick overview here. Just remember this graph. Sorry. So every 3 carbon dioxide that is incorporated, you have 6 glyceraldehyde that is being generated, out of which 1 goes into assimilation and 5 goes into regeneration. So now if you have 12 molecules, which is basically 3 into 4, so respectively it will be 5 into 4. So 20 glyceraldehyde, 3 phosphate. So you need to remember, not 4, 5 into 4, so 20. Okay. All right. Next one. Similar questions we have done also earlier. So which one of these is part of the payoff cycle? Payoff is with the second stage of glycolysis. So what you need to remember is there are 2 phases of preparatory phase and payoff phase. So you need to remember which ones are part of the second phase. See, so the ones which are involved are A, B, and D, inulase, pyruvate kinase, and phosphoglycerate mutase. And when you check so just be careful of these type of questions. This is one of the very common questions that keep coming up. All right. Next one. This is a very interesting question, and I wanted you to discuss with this because this is something that we discussed yesterday, right? Radio immunoassay. So look at the question here. It says serum isolated from a patient was subjected to radio immunoassay, and this was used to detect thyrotropin releasing hormone (TRH). Right. This is again from assignment 12. Since I cannot give you the answer, but I can. I chose to discuss this one question because this requires you to think. So it detects thyrotropin releasing hormone. So the signal detected was very weak. So basically, you are using RIA to detect TRH, but you are not getting it. So what could be the reason? It's already there in your assignment, so I'm pretty sure that you would have looked into it. B, which is very low? Absolutely. Because TRH is basically released from the hypothalamus, and this TRH releasing hormone goes to anterior pituitary, where it releases TSH, which is thyroid stimulating hormone, right? So that is the whole idea there. And now you're using RIA to basically detect the level. Now, RIA is a very sensitive assay this we discussed yesterday, but even then it is not able to detect any such level. So it means that the level of TRH in the serum itself is very low. It can't be very high. So the first option is incorrect. Third option says it's a lipid hormone that is not present in the hydrophilic serum, which is incorrect because it is a peptide hormone, so it is hydrophilic. D is it lipid hormone and it cannot bind to anti-TRH antibodies, which is incorrect because they are peptide hormone and they do bind to their antibodies. And E, TRH cannot be radiolabeled, so it cannot be detected, which is incorrect. We saw how thyroid was the one that was being used, right? Okay. Now this term here, P by O ratio for NADH. If you know what is P by O, this should be easy. These are some terms that are frequently used. And that's why I incorporated these questions just for you to know that you need to divide these. I could have put a lot of correct and incorrect questions, but those are redundant. You already understand the importance of those questions. .5. Fantastic. Exactly. So basically, P by O ratio, the name itself, it refers to ATP molecules that are synthesized. And what is P in that? It's basically phosphorylation per oxygen atom reduced. So oxidation. So P by oxidation, phosphorylation by oxidation. And that's what the term comes from, oxidative phosphorylation. So for NADH, if you are having, when you look into the ETC, you will see 10 protons are pumped out per NADH, but at the same time, 4 are moved inside for ATP generation, right? So overall, the P by A ratio is 10 by 4, which is 2.5. Yeah, fantastic. Next, this one. So basically here you have the nitrogen cycle, if you remember. And here you're given different bacteria here, right? So you need to give them the order on which nitrogen cycle works. So basically which one starts first, that bacteria comes first and then respectively the others. So which one fixes? So you need to remember the nitrogen cycle. What is the first step of nitrogen cycle? Fixing of nitrogen. Where does it start from? Remember all the nitrates, nitrites, atmospheric nitrogen, all these steps. EBAC. Okay, the answer is BECADF, and that's because of this. So you have to see, so what you need to remember is where are you starting from? So you're starting from azoto factor, which is fixing of nitrogen, and then you're moving from the nitrosomonas. Sorry. Then you are going to rhizobium. azotobacter rhizobium. Then you have C, which is your nitrosomonas here. Then you have A, which is your nitrobacter. And then you have the ones which are nitrospira and nitrococcus. Okay. All right. This one is about vitamins, which again comes from yesterday that we had discussed, and also from lipids. This is a, there are 3 more questions, this and 2 more questions, and then we can wrap up. Okay, let me give you the answers for this so that we can quickly go to the next one. So the first one is correct here. So your A, which is your vitamin E, that is called tocopherols. You will know these names. B is vitamin 3, D, which is cholecalciferol, which is also correct. We know this is how it is derived. C, vitamin K, is produced by intestinal bacteria. And ubiquinone, this has to be the easiest, and that was the option in every possible option there. So ubiquinone is lipophilic electron carrier, right? Okay. Second last question, which is the same. This is like a fill in the blanks question that we have done. I don't know how many times since childhood. So the first question is the measured difference in pH between stroma and the thylakoid lumen represents a large difference. So what is the pH of thylakoid lumen? Second one, which cycle is responsible for CO2 assimilation? That is just something that we have just done. And third is how many electrons are actually involved in the oxygen evolving complex that splits water. And the answers are there. So you just need to give me the option which is correct. D. Fantastic. So thylakoid lumen's pH is 5. Calvin cycle we have just discussed. That's for carbon assimilation. And 4 electrons, that is also we have something we looked into. 4 electrons are released when you have the splitting of water. And the last question, which brings us to the basics of Michaelis-Menten kinetics or the enzyme kinetics. We'll do this and then we will wrap it up. Which one of these is correct? Okay. A and C are correct. A and B and E are correct. I will see why. The first one is saturation effect occurs when there is no free enzyme to combine with substrate. Makes sense because if there is no enzyme, then you don't have any more kinetics. Next one, the overall rate depends on the Kcat, or basically the catalytic efficiency, which is correct. So the overall rate also depends on the total enzyme concentration. That both of them contribute, not just Kcat and enzyme to total enzyme concentration. Now this one, third one, the formation of enzyme substrate complex takes a long time, but the conversion of complex, which is ES, into product occurs quickly. It is incorrect because we have discussed, remember, formation of ES is fast, but the conversion of ES to product is slower, and that is mostly K2 is the rate limiting step. We have discussed this K2. The first one, E plus S to ES is K1, reverse is K minus 1. They are in equilibrium. But when you go to K2, which is ES to E plus P, that is K2. That's the rate of that reaction is K2, and that is slower. That is a rate limiting step. So rate, remember this, right? D is Km reflects the affinity of the enzyme for substrate under all conditions. This is incorrect. What is correct is the next one. Km is actually the concentration of the substrate at which V naught reaches half of Vmax. The data we know from the curve already. While D says that it is the affinity of the enzyme under all conditions. That is incorrect. It is apparent. We know it's a constant. It's apparent constant, and it is related to affinity, yes, but not under all the conditions. So that is a very over generalization, which is not correct, right? So generally, if you have to say about Km, you will reflect, if you will comment it on the basis of the concentration at which Vmax reaches, V naught reaches half of Vmax. Okay, right? It relates to affinity, yes, that's correct. That because when you look at Km and when you see the dissociation complex and all the other rates, you do understand the affinity of the enzyme towards the substrate, but it is not under all condition, and that is why it is incorrect, right? All right, so that brings us to the end. All the best for your exams if you're taking it. If not, all the best for your course if you are, whenever you are reading it again and doing anything with that in the future. Revise all the topics. The questions that we discussed today are the topics that are definitely very important. And keep reading, and all the very best for your future. Thank you so much for joining. It was very nice being with you people. Thank you. I think they will be generating it in the coming weeks because this is the last, like the courses are wrapped up now. So once every deadline and everything is met, all the assignments are completed and everything else is done, all the deadlines are met, is when the hall card will be released, and you know that you will get the notification for that as well, right? So yeah, that will come to you, don't worry. As